am shocked to see all of you here, actually, on Friday afternoon. So you guys are dedicated, right? So that is really awesome. And thank you for staying. So we are running about 10 minutes behind, so I'll try to get us going and keep us maybe see what I can do about getting us back on track. But I am, thank you to Paul for giving me a little bit of an out. I am not a virologist, I am an epidemiologist. So I'm gonna talk to you about a large-scale virology project that has a lot of great virologists on it and great collaborators, and some here, and existing and future and past collaborators. So hopefully what we're doing is generating a lot of material that a lot of you folks in the audience will have to keep going with. And part of what I do also is work with funding agencies and try to encourage them to release more funds for virologic work. So so we can talk about that later. That requires wine and a long weekend. All right. Not bribery, just that came out wrong, right? I really just mean just, you know, to be honest, it requires me to have one. Okay, so, wow. Anyway, back on track. I am a scientist, actually. But today I want to talk to you a little bit about shifting the disease paradigm. So probably, I think everybody knows we're going crazy. We're chasing Zika, we're chasing Ebola, we're chasing West Nile, if you were working then. I don't know how young all you are. But so we chase everything. We chase the last thing. And I guess I'm here to say let's go ahead and keep working hard on those things that are really serious and causing us trouble, but let's also work hard to get ahead of that curve, right? Let's stop chasing so much and think a little more proactively. Because as we see, there are probably some of the most brilliant scientists in the entire world working on things like influenza, and yet, when we have a new one, which we almost always do, right? Strain anyway, we are still trying to get a vaccine together pretty much after most of the world gets infected. So I'm going to talk to you a little bit about probably a kind of crazy scheme that is actually providing a little bit of proof of concept that we might be able to look at things in a little different way. So this is the crazy part. The U.S. Agency for International Development actually is responsible for putting a lot of vaccine out around the world. Not making the vaccine, but paying for it and delivering it to many less developed countries as part of the U.S. government's health diplomacy. And I think on some level, there are also great scientists within USAID, and they said, you know, are we doing the right thing here? Are we just band-aiding? Is this just diplomacy and not health? And so obviously, vaccine is needed, and it's not just that for everything, but for emerging infectious diseases, they're often too late. And so they put out a call about 7 years ago asking for teams to bid to try and stop emerging infectious diseases at their sources. And as Paul mentioned, usually we can track those back over history, and those are usually animal hosts. Not too shocking, not brilliant science, naive populations. When we're worried about humans, we're the naive population, are going to be more susceptible to severe disease for something we didn't evolve with that spills over to us. So a group of us decided to go ahead and bid for this project that USAID called Predict. Now, the exciting news is you get to try something new, try to go out from a One Health perspective, thinking about the people, but also thinking about the drivers, environmental drivers that might cause spillover, and the upstream hosts that might not get sick because they are actually the reservoir hosts, so they have evolved with those viruses, trying to look for those viruses that might become epidemic and pandemic. And so these are a group of partners that we've been working with, but we're working in 35 countries, or we have been working in 35 countries. So we work with governments and universities within all the countries where we work. Now, the bad news of that is that if you're really good at it and no viruses spill over, what happens? You're out. no funding for you, no more emerging infectious diseases. But wouldn't that be a great problem to have, right? Wouldn't that be a great thing? And I don't think any of us think we're going to solve this issue, but if we can help chip away at it. So here's kind of the background that I'm talking about. So we have, if you think about this as a regular epidemic curve, number of cases, right, over time. In the green, we have the potentially zoonotic viruses. We have all those viruses circulating out in primarily wildlife species. In the green, we're talking about wildlife. In the blue, it's domestic animals. And we know a lot more about what's in domestic animals because we've co-lived with them for so long. So it's these little things that are circulating in the green areas that might have spillover events to other species, also wildlife species, and you might see some die-offs, but you probably aren't going to pick much up there, okay? But then when it spills over into the blue, the domestic animals, or into the red, the people, we may see a spike of cases of disease, right? And so we want to know more so that we're ready, so that we could intervene or prevent those epidemic curves from getting so high. And to do that, we believe you need forecasting. So you need to model and think about where these viruses might occur and how they might occur and which ones. But you also need to be able to detect. And I think that may be the most practical place where we can have an influence or make a change in our lifetime, is make sure that people like you exist in all of the other places of the world, right? Where people can do this kind of work, can detect virus, do have laboratories, and can do diagnostics quickly, so that we don't have to see, like we did in West Africa, months go by before a proper diagnosis is made, okay? So that early detection is totally key. But early detection, when you don't know what you're looking for, you can't just roll out diagnostics to the world when you don't even know what viruses you might be looking for, what might be next. But the goal is to lower those curves, the red and the blue. All right, so we started and developed sort of building the plane while we were flying it kind of analogy, because who knows how to go look for things that you don't know exist and where to look for them, right? So we tried to use the best modeling we could, and we have several teams working on modeling to help us target where we might go look, okay? And all models, apologies, I like to model. If you like to do mathematical modeling, they're all bad, right? They're all bad, and we make them better. That's science. And we keep making them better and making them better until they're great and we don't need them anymore, and the next ones that we do need are bad. But anyway, so that's just the way it works. But it's helpful because we can target resources. I think that's the best use. So we wanted to target using the best available data that the world had to offer, as well as the best information from the ground and our partners, to look at the right interfaces where animals and people were coming together and facilitating viral spillover from one host to the other. And then we had to target species. We couldn't just look in every species. So we needed to look and find what might be the most likely species for the next viral spillover event. Then the pathogen diagnostics and discovery, as you've been talking about a lot today, and what I do believe is critical as far as capacity building to prevent these horrible epidemics and potentially pandemics from happening in the future. And we believe we should be able to put together and look using platforms that identify both known and novel potential pathogens, okay? Known pathogens and novel potential pathogens in the same format. But I'm talking about the least developed places in the world, okay? So we don't have metagenomics, high throughput sequencing available to us in these places. Some of the places where we work didn't have a PCR machine in the country, okay, when we started 7 years ago. Okay, so that's what I'm talking about. How do you do that? How do you make that work happen, okay? And then you have to share the virus, and you have to get the information out to the public, and you have to get it into the hands of the people who can make good decisions with it. So this is just an example of one of our modeling activities to figure out where to focus, what kind of targets, both in the field. These are all the interfaces. So the red dots are viruses, all the zoonotic viruses where we have documentation of how they spilled over from animals to people. And the bigger the red dot, the more interfaces it's connected to. So indirect contact and direct contact are the blue and green. green, and then vector-borne is the yellow. We use this for a couple different ways. We used it to target the interfaces. We also used it to target viral families that we might be looking for as far as the more zoonotic potential. And it also helped us because our colleagues that we're working with in the US government were also doing a lot of good work. We didn't want to overlap too much. And so a huge amount of effort for decades, great work has been going on with CDC, but it tends to be more on the vector-borne side because the disease burden is so high there. So it made sense with USAID funds to focus on sort of this hot mess over here. And so that's what we did. Okay, so speaking of hot messes, this is a bat guano farm. So you won't find that in the previous slide with all of those interfaces because there aren't many documented spillover events that we can detect in bat guano farms because they haven't been that popular and they happen to be in places where people don't write a lot of papers and people aren't looking for spillover events, right? And so, but if you build a nice bat condo and you're in Southeast Asia or some other places, the bats will come and visit you and they will give you some nice fertilizer, all right? But that was becoming more and more one of those interfaces that we had to identify on the fly as we went, working with our local teams. So in all of the countries where we work with teams implementing partners from the countries. It's not just UC Davis and all these global partners zooming in and saying, we know what to do, we're gonna protect you, right? It's not that sort of colonial kind of science. We're doing it in partnership with the countries where we work. So again, looking for those high-risk interfaces, but not forgetting the large-scale ecological drivers. So going upstream and continuing to look at what might be driving that increased contact or the ecological disturbance that sets off immune systems so that susceptibility changes or shedding changes in the host. So we put all those into our magic black boxes as well. So what about proof of concept? So I'm gonna just walk really quickly as I'm just screaming at you in a rapid fire pace, but try to get us, I'm not gonna get us back on time. Anyway, okay, so what could we do? So I'm talking about countries where no viral surveillance was really going on, except maybe WHO flu surveillance, all right, at only certain sorry sites, and absolutely no real wildlife work to speak of, like systematically through the government. And in the first 20 countries where we started over 5 years, we were able to safely access and sample 56,000 animals. That's a monkey in a cage there. And we're a no-kill project, so we're all conservation oriented, and we're just as interested in the viruses that might be spilling over from the people into the monkeys as we are from the monkeys into the people. We did, with our modeling and targeting, we were able to hone down to the most likely suspects, given what's emerging now and where we understand viral diversity to be the non-human primates, bats and rodents. And so that was our focus, though in certain instances where there were interesting interfaces, intimate contact with other species, we looked at those as well. And then we did develop and operationalize a diagnostic platform, again, in some of these least developed places in the world. And we used consensus PCR. So we went back to basics, conventional PCR, consensus PCR, with then cloning and sequencing, so that we could pick up known and novel viruses. And we did that by targeting all of the viral families that we could identify that we thought were likely to go epidemic and pandemic from those families where we've seen an epidemic in the past. So it's not perfect. We did do some high throughput sequencing side by side, both to compare our techniques as far as what we discovered and sensitivity, all of those pieces. But we actually were getting more virus and more interesting virus, frankly, from the consensus PCR platforms that we could run in the country. And the important thing there is that then those countries can detect, diagnose these viruses when they emerge. So not necessarily be doing surveillance in advance for a whole bunch of things that they don't know are pathogenic. They don't have the resources. We don't have the resources in this country to do that, right? But they could detect something new should they have unusual cases. we had to develop universal control material, plasmid-based controls that allowed us both to detect contamination in those laboratories. Happened a lot. We had a lot of training to do, a lot of cleaning up to do, but also because we can't send around the most dangerous pathogens in the world for positive control material, right? So we have to be safe in these situations. And so what did we do? We ran over 400,000 diagnostic tests, and in the first 5 years, we detected about 1,000 viruses. We're way over 1,000 new viruses now, but about 812 novel viruses in that first 5 years, and about 150 known sort of bad actors, known pathogens, using that consensus PCR platform. So we were very excited about that because it was operationalizable. That's a big word that you never need to use unless you work with US government people. Okay, say it 3 times fast. So anyway, we're really excited, but we found things in those categories that were scary. But then what? Then what do you do with it? So if you discovered 100 novel coronaviruses, right? What do you say about that? What do you tell the governments? What do you say responsibly? Which one's going to be a nasty bit, right? And so that's where we are now. We're working on that viral ranking strategy, and I'll talk to you a bit about that as we go. So we also, back to the Bat Guano Farm, we also started to find in real time things like the most closely related MERS-like viruses, right? In places like Thailand in this particular paper. But during the MERS outbreak, we had found a MERS-like coronavirus that wasn't out in the public domain yet in Mexico. So we were able to get with the Saudi government, along with our colleagues who were being asked to go and investigate this new disease and say, all right, check out bats, check out these different kinds of locations and interfaces for your surveillance. So able to help participate and target real time. Probably more interesting for this audience, we could start to get the first estimates of total viral diversity in species because we sampled species intensively. So we could start to look, for example, at certain species of bats and start to get the saturation curves for how many viruses, how many animals do we have to sample in how many locations to discover everything they have. Now, of course, we don't know all of what they have, but we do start to see asymptotically that we're getting to diminishing returns. And this graph shows us that at about 80% of that viral curve, you know, we get 80% of the viruses with about half the effort, as if we kept on going and spent a lot more money to get the super rare ones. That's also good for, you know, figuring out how to develop these kinds of platforms, because the super rare ones are going to be super rarely transmitted as well, right? So the more rare they are, the less I feel like we really need to discover them. It's still important and interesting, but maybe not worth the cash, right? So this also helps just on simple things like your IACOCs and IRBs and all of those things that ask you to have rigorous sample size estimates when there was no way to do that before because you didn't know what you were looking for or how many might be out there. So starting to contribute to that. This is a global hotspot map for viral emerging disease, and now I'm happy to say that we can make it better, okay? So that one's about 10 years old. We're constantly improving that and figuring out better mechanisms, better variables to include in our models, things that we weren't necessarily looking at before, like the changeover of pasture land being super important. We knew intellectually changeover of forest might be important, but changeover of pasture also super important. So we can start to hone that map so it's not so red, so that we can target our efforts even better. In this program, in the first 5 years, we assisted 10 country governments in responding to 23 different outbreaks. And we made sure that every viral finding that we had was distributed to all of the ministries in a One Health approach. So not just the Ministry of Health if we were worried about human samples, but the Ministry of Environment and the Ministry of Agriculture, got them sitting and talking together and had some really excellent outcomes in the way of outbreak response, in bringing those different sectors together so that they would be thinking again upstream about the risks and how exposure was happening. happening. So now we get that information after the government's approved release of the data, which has been pretty easy. We thought it was going to be the hardest thing to do in the project. Now we're allowed to get that data right out, and we have our own interface on HealthMap that you can get to through HealthMap, but it's probably easier if you go to predict.global, and you can go right in through our web portal there. You can see all of the places we've sampled. You can see all of the viruses we found. And if they're already, the whole sequence is published in GenBank, you can link straight out to GenBank there. Not all of them are there yet, though, but we're slowly getting them into a GenBank system. All right, so what's next? How do we decide which of these viruses are the important ones? Which are the ones that need NIH funding for intensive studies for characterization and follow-up? We have the ability to do like full genome sequencing and characterize them to some level, but we're not necessarily able to go the distance like the things that you guys can do. And so, so we're working on trying to come up with a ranking system, but instead of just going virologically, because that doesn't seem to work, we're also looking epidemiologically and ecologically. So at those interface issues where people are likely to come into contact, what sort of transmission interfaces and how important are those transmission interfaces, including that into our system of viral ranking. Because we know that probabilities are showing us that even though near neighbor sounds like a good hypothesis, it's not really showing us what we need to know. So how do we choose the right viruses to go in and do more receptor work and do more detailed studies? And so we're going to need your help, and we're going to throw out some hypotheses, and we're going to get a lot of criticism, and that's okay. More wine. Okay. All right. So now in EPT2, our new program, we're in the second 5-year phase of this project. It's not a small project, $175 million project over 10 years. And in this 5-year phase, we're actually now not just looking primarily in the wildlife for those things that might spill over, but now we are actually targeting detecting viral sharing. So not transmission, we don't know which direction, but we're looking in people, wildlife, and the livestock at those identified interfaces in 31 countries. And hopefully that will help us, again, discover more virus, but also more drivers that we can use for mitigation. So that means we have a heavy focus on behavior. So where does that leave us? What do we know now? Well, we know that there are a lot of drivers like the rapidly increasing human population, and what that means is expanding into more wild areas, using the land and animals differently, using resources differently that upsets the ecological balance that might lead to disease expression. And so that contact rate and those interactions are probably leading partially to what we're seeing and experiencing as increase in emerging infectious diseases. And so just as a little precursor of potential things that you might be the ones to follow up on is that we do think, given that in just 5 years we were able to discover 1,000 viruses with basically starting from absolutely from scratch and training people to be safe for themselves and the animals all the way to laboratory biosafety, all of that. We do think that we can hone this and target this effort to be able to take it worldwide. And we've identified 23 viral families that we think are of most concern. That helps us narrow our targets of where we need to look. Again, I think the best platform when we're working in middle and less developed countries is to think about a combination of this consensus PCR inexpensive with a high throughput sequencing to make sure we're getting good work done. So we can also narrow our search by getting down to just those hosts that we think are the most likely targets. So if we narrow by viral family and by hosts, and we use those viral diversity curves that I mentioned before, we can get it down to, we think there's about 1.3 million viruses out there that we should discover. We should know about, we should be ready for. I'm not just saying that we should go out and do surveillance for all of those viruses, but we should have them on the list. People should be able to detect them. If people are sick, animals are sick, we should be able to pick those up. And what does that cost? Well, really only about less than half of those are likely to actually be able to infect humans. Okay, so there's probably about 500,000 that we would then, have a list that we'd have to hone down to the most likely suspects. But to go out and do that effort would cost about $3.5 billion, a little less than $3.5 billion. And we spend that routinely on emerging infectious diseases. Okay, so the World Bank estimates that we could save $6.7 billion a year if we knew better how to prevent zoonotic disease. All right, so we're talking about a lot of money, right? Everybody'd like to have $3.5 billion in your lab, right? But it's a lot of money, but it's a fraction of what the world spends on chasing these things. So that's my story, and sorry I didn't talk about receptors or enzymes or anything else. You might get that in the last talk. But this is really where I hope we're headed. So early disease detection and hopefully improved response. Thanks.
I had a question. You talked a lot about modeling and sort of thinking about who's at highest risk for viruses to follow up on. And I'm wondering if you've done any sort of backward simulation looking at Zika virus is a great example, right? Here's something that's been around since the late 40s. Or
forever.
Yeah. And here we are in 2016, and it seems to be a pretty significant crisis.
Yeah.
Is that something, can you say a little bit about whether the algorithms or the types of models that you've used, have you looked at that or done that kind of simulation, looked backwards at prior emerging agents and been able to discover them?
Yeah, absolutely. So, I guess we've looked back at every zoonotic virus we can find in history, and then in the 1990 to 2010 period, really intensively at every spillover event to try and characterize what were the circumstances ecologically, human-wise, all the circumstances around that to help us with forecasting. The question with Zika, I think what's really interesting and what hasn't been done, and shame on us as a community or whatever, is that we tend to talk about emerging diseases because we all want to discover the next new one, right? But we leave the really interesting, potentially important ones that might change a little because they get into a different kind of naive host population or because they mutate or other things happen. And I think that's a problem. I think that we haven't been doing that, and we're trying to do that now is the answer. So with MERS, for sure, that work, because we've known about it just a little bit longer, we're doing better work there. But I do think that it's critical to think about emerging diseases being emerging into new areas and new hosts, not just a brand new thing that's new to be discovered.
Hi, great talk, and I applaud your efforts for working abroad and starting these surveillance initiatives. So even from my own experience working abroad in Peru, one of the major challenges was you have a group of very inspired individuals who will go down and try to implement these surveillance activities, but then you find out that there is a true lack of infrastructure in these developing nations. Mm-hmm. And one of the challenges was once these investigators leave, often these programs just do not continue because of lack of funds or just the lack of infrastructure. Yeah. So I'm just wondering if you could just comment on a little bit of the solutions you found to working around that. Does that mean that we need to build more infrastructure? Does that mean we need more long-term programs to continue? Or do we need to engage the local governments and communities to actually bring about true change?
Yeah, all three of those things. I think we do need more long-term programs. We have some good examples of successes with that, with like PEPFAR HIV programs, things like that, where there were long-term efforts. We need to not do like what Congress and the House are thinking of doing right now, like we're going to invest in Ebola and then tomorrow we're going to take it away and we're going to move it to Zika and then tomorrow we're going to take, so you just get started, then you pull it away. It's completely distracting and you can't do anything rigorous in that kind of scenario. So we need to have horizons and set them and stick with them and then extend them if necessary, but don't cut them back because, well, it's been 6 months and I don't see anything happen. Well, in these places where there's no infrastructure, just getting a contract with the federal government is not very easy, right? So we do need that. I believe we need capacity building at the most basic level, and that means find those folks that want to do this forever and train them and give them the equipment and the ability and the collaborations. So that's more like the university twinning and working with those universities so that the graduate student or the postdoc you're training there is their professor next time and is out there doing this work for their career. Then it will continue. And then, again, just the lab capacity strengthening.
One over here. Is this on? Yeah. Hey, Jonna.
Hey.
Great. It was great
Wanna
to have
go
the opportunity to hear you lay it all out there. So thank you. So mine is also kind of a non-scientific question, but you talked about the detection and the early forecasting, and then of course there's the infrastructure issues, and then you've got the rapid response bit, which of course is political, and it's like all sorts of things which are not necessarily predictable or scientifically driven, and yet you have experience now working with 23 outbreaks in 10 countries. How has that part of it been? Because of course doing something about it and actually bringing that curve down is going to require not just you but the government action, and
sure.
Yeah, that's always tricky.
I think it's been incredibly daunting and then satisfying. So and the stories differ from country to country, but some of the really great ones, my Ugandan colleagues always say, come to Uganda, we are rich with viral diversity. Anything you want to discover, we probably have it here. So Zika included, right, where we first found Zika, so the global we. So for example, really quickly in Uganda, when we started working there, their government could not release funds for an outbreak until they had a diagnosis, so they knew which funds to release. So if they had an undiagnosed event, which I'm sorry, but almost all of them are, especially at the beginning, they have to be, right? Then they couldn't release funds until basically an external entity came in and diagnosed it, and then they could release funds from the government, then they could respond. That has completely gone away. So in about a 4-year period, we saw their country go from that to having a task force, it's called the Zoonotic Disease Task Force, but it's not necessarily just for zoonoses. It's basically an undiagnosed disease task force that meets now within 24 hours of one undiagnosed case if they find out about it. And they have on those teams, our predict country teams are on those teams and have developed the SOPs for animal sampling, environmental sampling, the instant they have an undiagnosed case of disease. So within 24 hours, people are going to the villages. So that's shocking and great, right? And then other places it's not. And then there's, the story of 2 Ebolas. So in DRC, where they've known about Ebola for a long time, and our teams were there and helping, and a diagnosis was made within just days of the first cases in very remote areas at the same time as the West Africa outbreak was ongoing. And the government immediately jumped in, put some transportation travel constraints in place, and less than 100 people died. We didn't have the West Africa situation. So that's a combination of education, outreach, and capacity.
So obviously this is a very important area of research and trying to come up with some kind of rationale of how to anticipate perhaps what's the best way to use resources. But just knowing what is out there is not going to do it.
Right.
You need to apply some kind of filter to these are the most likely pathogens, viruses, whatever, that will jump into human populations. And I didn't get really what is the criteria that you guys are handling at the moment, or is this something in progress?
It's definitely in progress. We have, I'll tell you where the progress is. We have about 8 different mathematical models that all spit out different probabilities. I'm trying to link them, and some of them are ecological and epidemiological, and some of them are virological. That said, we're going to miss a lot, because when we're talking about new viruses or viral families that haven't been investigated a lot, then there's so much information that's yet to be known. We're going to not do it right. With those 1,000 viruses, we're trying to actually just narrow it to a list of 100. So we're trying to take it about 10% down to say these 100 deserve more work, laboratory work, all sorts of work. But I can't give you that math today, but that is a work in progress. And I think, again, it's going to be a straw man for the community. It's going to be a straw man of this is the best we can do with all the information that we know. From our perspective, throw it out to the scientific community. And again, be okay with being wrong and let people improve that as they go.